Hello, we are group C2.2, and we're going to be talking about dopaminergic circuitry and signaling in Drosophila melanogaster. And more specifically, we're going to be investigating sweet taste appetitive behavior through neuroanatomical and behavioral assays. So, appetite and hunger are natural internal states elicited by bodily needs for nutrients and energy. They are encoded by neuromodulators, which interact to form an intricate regulatory network that gives rise to coherent feeding and foraging behavior. In Drosophila, dopamine receptors are involved in controlling food preferences. Specifically, dopaminergic neurons, abbreviated as DANs, in the protocerebral anterior medial or PAM cluster respond to the sweet sensory properties to signal sugar reward. Previous research has indicated that silencing DANs decreases the proboscis extension reflex, or PER, of starved flies to sucrose, while activating DANs elevates PER to sucrose. And PER is a simple taste behavior that initiates the feeding process. However, fly appetitive responses to sucrose when dopaminergic levels are upregulated and downregulated by different assays are still unclear. But based on this knowledge from prior research, we hypothesize that the fly lines expressing dopaminergic activation will lead to offspring with a higher attraction to sucrose feeding, while silencing DANs will lead to no preference for food choice. Drosophila melanogaster are favored biological model systems to study the mechanisms of dopaminergic neurotransmission and its related behaviors, because they are they have a relative human genetic resemblance. 75% of human illness genes and over 50% of human disease proteins are observed in Drosophila. Thus, they can be genetically tractable and modifiable through the GAL4 UAS system to examine the neurological pathways that modulate function and behavior. Our research may suggest new correlations between dopaminergic circuits in the Drosophila mushroom body and patterns of how taste information is processed to affect both feeding and appetite across genetically related species, including humans.
In this experiment, we had three aims. The first aim was a neuroanatomical assay in which we mapped the neuroanatomical distribution and function of dopaminergic neurons, DANs, in the fly brain. Aim 2 was a thermogenic behavioral assay where we examined and tested the functional role of DAN activation on fly appetite. And aim 3 was an optogenetic behavioral assay where we examined and tested the functional role of DAN silencing on fly appetite. To perform these assays, we requested the generation of our required cell lines from the lab. In all conditions, we used GAL4 line flies expressing dopamine and manipulated the UAS lines for each experiment. Under all experimental conditions, flies that are used have been isolated as pupae until adults. The flies are kept in individual vials containing yeast food and were then treated differently under specific conditions for each aim, which we will discuss next. For our first aim, which is our neuroanatomical assay, we will cross GAL4 8848 with UAS 8515. With this, we aim to do a dissection of the brain and then image it with fluorescent microscopy. Through this, we expect to see fluorescent staining of DANs, most concentrated in PAM cluster. For our second aim, the thermogenic behavioral assay, we will cross GAL4 8848 with UAS 26263. We aim to thermogenically activate dopaminergic neurons at 26 degrees Celsius. Through this, we expect to see the fly activity per DAN vial to be concentrated at the vial end with 0.3 molar sucrose. For our third aim, our optogenetic behavioral assay, we will cross GAL4 8848 with UAS 41752. where we aim to optogenetically silence stands using yellow light that activates halorhodopsin to hyperpolarize stands. Through this approach, we expect to see the fly activity per dam vial to be equally distributed between both food groups.
During week 1, we will cross fly lines GAL4-8848 with UAS 26263. In week 2, we will cross fly lines GAL4-8848 with UAS 41753. And during week 3, we will cross fly lines GAL4-8848 with UAS 8515. If any issues arise, we will recross whichever lines have issues. For our first experiment, we decided to map the neuroanatomical distribution and function of DANs in the fly brain. This study used an anatomical approach utilizing fluorescence to highlight the location of dopaminergic neurons in the fly brain. We crossed the dopaminergic GAL4 line with the UAS line for GFP to use green fluorescence to map the distribution of DANs in the fly brain. By using an anatomical approach, we will examine each of the flies with GAL4-8848, which is the dopaminergic GAL4 line, crossed with UAS line 8515, which is for EGFP. This cross will allow us to target dopaminergic neurons and use green fluorescent light to map out the location of DANs. In order to determine the final mapping of the dopaminergic neurons, we will need to dissect and process the fly brains under a microscope and shine green fluorescent light to determine the location of the DANs. We will examine at least one fly brain from this cross to determine the expression of GFP in the fly brains, allowing us to determine whether these DANs can be mapped, ensuring that dopaminergic neurons are being targeted. Flies will be raised at about 20 degrees Celsius on standard food media, which is yeast-based, and dissected at least 2 days after eclosion. After dissection, brains will be washed and fixed onto slides for imaging and analysis. In our second experiment, we examined and tested the functional role of DAN activation on fly appetite. This experiment directly manipulates DAN activation using genetic tools to modify DAN activity and release during fly isolation. The manipulation of DANs will modify fly behavior, allowing the determination of whether the presence or absence of sucrose is more preferential for activated dopaminergic isolated flies. We used trip activation of dopaminergic neurons to determine fly behavior in regards to food choice. Flies will be raised and kept at 20 degrees Celsius until 24 hours before experimentation, where flies will be moved to warmer conditions of 26 degrees Celsius. We will put food on both sides of the tube inserted in the Drosophila activity monitor, or the DAM, and one end will have regular yeast-based food while the other will have sucrose food. We will run the experiment for 24 hours in the dark, monitoring the frequency and proportion of time the fly spends at each end of the tube. In our last experiment, we examined and tested the functional role of DAN silencing on fly appetite. These studies will directly manipulate DAN silencing using genetic tools to modify DAN activity during fly isolation. The manipulation of DANs will modify fly behavior, allowing the determination of whether the presence or absence of sucrose is more preferential to silence dopaminergic isolated flies. In prior studies, halorhodopsin is often used to silence neurons with the use of yellow light. We will be using YFP tagged yellow light activated halorhodopsin to silence dopaminergic neurons, which will determine fly behavior in regard to food choice. Similar to experiment 2, flies will be raised and kept in room temperature conditions of 20 degrees Celsius until 24 hours before experimentation, when flies will be moved into warmer conditions of 26 degrees Celsius. We will put food on both sides of the tube inserted in the DAM monitor, and one end will have regular yeast-based food while the other will have sucrose food. We will also run this experiment for 24 hours with yellow light shining on the DAM, monitoring the frequency and proportion of time the fly spends at each end of the tube.
In flies, the GAL4 UAS system is a well-known and powerful approach used to genetically manipulate and map the cellular function of Drosophila. In this system, the GAL4 gene encoding for a yeast transcription activator protein is controlled by an enhancer region that will express the protein in desired cells in one of the flies. These are then crossed with a second line of flies containing the upstream activator sequence, also known as UAS. GAL4 binds and activates gene transcription, allowing the desired protein to be expressed. The expression of yeast genetic tools does not influence other cellular processes in Drosophila and is not endogenously present in flies. Therefore, these tools are ideal to be used in the examination of gene expression in Drosophila and ensure the selectivity of manipulation. A GAL4 genetic line must be crossed with a UAS genetic line in order to drive expression. There are typically no interactions or side effects in the insects carrying uniquely UAS or GAL4, allowing for safe amplification of the lines before generating experimental crosses. We will employ the GAL4 UAS system and use 3 founder lines to generate 3 experimental crosses as seen in Figure 1. These genetic lines will be acquired from the Bloomington Drosophila Stock Center. We will use GAL4 line 8848, which expresses GAL4 in dopaminergic cells. A mapping of dopaminergic neurons in the fly brain can also be seen in Figure 2, labeling adult fly DANs tagged with GFP obtained through the virtual fly database. We will combine GAL4 line 8848 with 3 different UAS lines. For the anatomical assay, we will use UAS line 8515, which drives the expression of EGFP under the control of UAS and allows for anatomical analysis. We will use UAS line 26263, which drives the expression of TRPA1 under the UAS promoter. This allows for transient activation of the targeted neurons at a high temperature of 29 degrees Celsius. Lastly, for behavioral assay and dopamine inactivation, we will cross the GAL4 line with UAS line 41753, which expresses a YFP tagged light activated halorhodopsin chloride channel under UAS control for cell hyperpolarization. Using an optogenetic approach, we can conduct a behavior analysis to study neural mechanisms underlying appetite in relation to dopamine levels.
So as for our results, for the neuroanatomical assay in experiment 1, we were unable to obtain a proper fluorescence microscopy image of our EGFP brain. We used the image from the virtual fly database to represent this experiment, which demonstrates the expected results of the activation of the dopaminergic neurons in the PAM-DAN cluster. In a future experiment, we could perhaps attempt to redo this neuroanatomical cross, obtain a better dissected brain, and produce a successful microscopy image of it. For the behavioral assays in experiments 1 and 2, we were able to obtain the data from the dam and create a graphical representation as shown here. Vials 4 and 5 contain the flies from cross number 2 with DAN activation, and we observed patterns of higher sucrose feeding as compared to normal yeast food, as the frequency and proportion of time that they spent at the end of the vial with the sucrose was larger than the end of the vial with the normal yeast food. Vials 1, 2, and 3 contained the flies from cross number 3 with DAN silencing, and we observed patterns of relatively equally distributed feeding between both sucrose and the normal yeast food. The frequency and proportion of time that they spent at each end of the vial was more or less equal, contrastive to vials 4 and 5. This reinforces the previous research from related studies, as well as our hypothesis that DAN activation will lead to a greater preferential appetite for sucrose, while DAN silencing will not lead to any food preference. Thank you.